Now for the next talk, we have a presentation on trachea and esophagus tissue engineering, and I think there's been a change in speaker. Postdoctoral researcher Philipp Jungblut, who's going to be speaking. Okay.
So thank you very much. Does it work? On behalf of my chief, Paolo Macchiarini, I have to apologize that he could not join this session and could not give the talk, so I'm the only thing that you get right now. But I try my best. We're working together since 10 years on the same things in Barcelona and now in Stockholm, so I guess I can tell you some things about it. So we work with the tissue engineering of the airways, mainly of the trachea, and we are developing both an in vitro method and an in vivo method. So I would like to introduce you to this topic. First, one has to ask which kind of patient actually need an engineered trachea. So there are a lot of different kinds of diseases affecting the trachea, both benign and malignant diseases, which could be probably cured by a simple resection and, by a simple resection and a re-anastomosis of the remained trachea. So you can do this procedure until a total affected length of 50 to 60% of the adult trachea. After that, you have no connective tissue, no reconstructive tissue to do this procedure, and the tension on the anastomosis would be too high. So what we need is an alternative to this resection and re-anastomosis, and common and usual other transplantation is actually not feasible due to the lack of vascularization in the trachea. So you have no big vessels that you can transplant, as it's the case in other organs. So it's technically very difficult if you want to do it because of the lack of vascularization. And in addition to that, the patient, of course need immunosuppression lifelong, and this would require, that would represent a very, very significant impact on quality of life. So we need an alternative, a safe alternative for these patients. So in 2008, we came up with this method. It's actually the classic tissue engineering method of using a scaffold, seeding with cells, and putting everything together in a bioreactor and transplant into a patient. What we learned from this case was that the most important in the beginning is that you have a non-immunogenic graft, a non-immunogenic scaffold where you can seed your cells on. So we used a donor trachea and we decellularized it, and then we checked for this major histocompatibility complex 1 and 2 to be sure that this is a non-immunogenic scaffold, non-immunogenic tissue that could be transplanted thereafter without the use of immunosuppression. What we also saw in this decellularized matrix is that there are still some pro-angiogenic factors inside the matrix. And this is very interesting and very important, actually, if you transplant this tissue later into in vivo conditions. We put it into chicken eggs, and we saw a significant increase of new blood vessels that are spread out from this decellularized trachea. We also analyzed the extracellular matrix. I guess on Wednesday, on this tissue engineering day, you heard a lot of the extracellular matrix, which is very important for cell engraftment, cell adhesion, differentiation, and so on and so forth. So we have all the important compartments of the extracellular matrix in the decellularized trachea. What we learned from the in vivo studies is that if you just seed, for instance, as you can see here, if you just seed the external surface of the graft, you have a very healthy external part, and it's very strong, the trachea, and you have no collapse, but you have a contamination, very fast contamination with bacteria and fungi. The opposite is when you just seed the internal surface, you have a healthy epithelium, but you have a collapse when the animals, this is an animal study by the way, sorry, in pigs, when the animals start to breathe, the trachea collapse due to a lack of strength, due to a lack of mechanical properties. So just if you see both the external and the internal surface of the graft, the animals can survive and the graft can survive and can really replace the trachea. What we also saw in our experiments is that the cells that you see that in vitro, they do what they want in vivo condition. They just move through the wall and they move through different oxygenation concentration. So at this point, we get a little bit upset about our whole in vitro method and protocol, and we said, okay, why are we dealing with all this long-lasting in vitro culture, bioreactor, and the big costs? So why are we not moving into in vivo conditions? And then we make a change from science into medicine, actually. And we took the bone marrow on the operation day and we put it directly back into the animal or into the patient later on. So we used also a donor scaffold. We decellularized it as we did before. Actually, you can store the decellularized trachea for around 1 year without losing any characteristics. Then thereafter, we isolated the stem cells, or the progenitors, let's say. We took a bone marrow aspiration, we isolated mononuclear cells, and then we seeded them intraoperatively in the theater on the scaffold. Then, this is very important, as we saw later, and as we learned in the very recent study, this boosting factor. This is very important. I will explain which kind of boosting factor we are using. And then we assumed this in vivo differentiation and engraftment of the cells. As I explained, we took the bone marrow aspiration. We use an automatic device which is isolating the mononuclear cells from the bone marrow fraction, and then we use for the internal surface. We use a biopsy from the nasal septum. We isolated epithelial cells. We mixed it up with autologous from the animal, from the patient, autologous plasma, and so we obtain like a gel, and we can cover the internal surface very gentle and very easy without any problems. And the cells stick in the plasma, and they have everything they need actually. They stick. They have all the necessities to engraft and to attach to the scaffold. The same thing we do with the external surface, and then we injected the boosting factors in the graft's wall, and then transplanted directly back into the animal or in the human being. The boosting factors that we use is first of all this GCSF. This is known as a pharmaceutical treatment to mobilize progenitor cells, especially endothelial cells. We also learned that this is not always the case. It depends which kind of pretreatment the patient had. We will show that very soon in a manuscript. We're still working on it, but it's looking very promising that we can show that this is not anymore the case, that just the endothelial cells are mobilized. We also use the erythropoietin, and we use it in a very high level, let's say for 2 weeks. If you use erythropoietin on a level like 100 units per kilogram, as it's used in the clinic, you just affect the erythropoiesis and you just enhance the number of red blood cells. But if you use it in a higher level, like more than 500 units per kilogram, then this erythropoietin binds to the APO receptor and to a beta receptor, let's say. It's a tissue protective receptor. It's a dimer of the usual erythropoietin receptor, which is expressed in all the cells, in all the tissue actually. And we saw that this binding has a positive effect on inflammation and apoptosis. So apoptosis is reduced by several pathways that are activated. I will show it later in a figure. And then finally we use this transforming growth factor. This is one of the factors that is involved in the differentiation of chondrocytes, both in vitro and in vivo as well. In vitro you usually have to add several factors, not only beta-TGF. You need several other factors. But in vivo, we saw that just mixing it up with the plasma and then put it into the in vivo conditions, this is enough to differentiate stem cells into chondrocytes. Now, what we observed after the transplantation, we took blood samples to see first how the hematocrit, is there any event of thromboembolic happening or something like that, which can of course happen if you use such a high level of erythropoietin, and then we isolated the white blood cells. And we looked for them, and we looked at the development. And one can say, oh, the increase of white blood cells is just an infection, or it's just an inflammatory response due to the operation. But you would expect the maximum, the peak, like on the third or on the second day after such a huge operation, but not on the fourth and not sixth and not on the 10th. That is here the case. So, but to be sure, we not just count the white blood cells, we also put them in culture. And what we saw is that we can, based on the in culture cells, we can isolate endothelial progenitor cells, and they also increase the number of capillary-like tubulars on this matrigeo. We also saw that we can isolate mesenchymal stem cells or mesenchymal-like cells. I mean, mesenchymal stem cells is very difficult to say right now. But we are just able, so far we were just able to fax these cells after in vitro culture. We were not able to isolate them from the peripheral blood directly by faxing. But we were able to see and to fax these cells after some weeks of in vitro culture. We were able to differentiate them into different and distinct cell types, which are known as stem cell-like differentiation cells. And then so this was a part to see the increase of white blood cells is not an inflammation, not just only an inflammation response to the operation, but there are specific cells from the bone marrow mobilized, which we can detect later on, and which should have a positive effect by homing to the graft, by reducing the inflammation, and also by active remodeling of the graft. The EPO that I explained, we used in a higher level. So this is a clinical used, the daily used clinical dose of EPO to increase erythropoiesis in patients with anemia, for instance. But if you use this 500 units per kilogram or more than 500 units per kilogram, then the EPO can bind. So there's a different affinity to these two receptors. And if you use this high level, then the EPO can bind to this tissue protective effector, and via several pathways, you active some anti-apoptotic genes, especially this BCL2L, and you inhibit the apoptosis of all the cells, especially of the mobilized stem cells. That is the theory of all this. We also can detect this better receptor, this tissue protective receptor on the trachea, in the implanted graft actually also, and it was increased after increasing the level of EPO postoperatively. What we saw when taking biopsies, this is normal trachea. This is a bionic, intraoperative designed and engineered trachea. We saw after 7 days it doesn't look very healthy. It doesn't look like a real trachea. But after 30 days, we had nearly a complete native trachea. The only difference to a native trachea is that we have a double cartilage band. So far we could not explain it, but right now we're doing some cell tracking experiments to see if this just the cells that are seeded intraoperatively, or is this cartilage ring the seeded mesenchymal or mononuclear cells that are differentiated in chronocytes. So we are just evaluating what happened in this region. But so far there's no negative effect on this double band. It's actually more strength, this engineered graft, than the native one. These are the mechanical properties. As I said, there are no significant statistical differences of the mechanical properties of the engineered graft to the native. So our clinical experience so far, we transplanted 9 patients with this method. We recently transplanted another patient, but with another method, which I will explain in the next slide. The indications were very different, as I said, both benign and malignant. We replaced the entire or just the main bronchi in these patients. We had the same findings, that no epithelium after 7 days, but we had a very good looking and nearly native trachea wall after 1 month in these patients. The intermediate results are like that we have a completely native looking epithelial ciliated cells after 1 to 6 months. We have a basal membrane 2 months after the transplantation. even this one is from an in vivo engineered trachea. So we applied already both methods in the clinic, in patients, and looks quite good. What we have in the late outcome so far is we had three patients with an anastomotic malathea in the distal part of the engineered graft. These patients received a biodegradable stent which disappeared after 2 months, and all of them, they will come in the next 2 to 3 months for follow-up. And then we have to decide if they need again a biodegradable stent, but so far it looks quite good that the trachea is completely open and they do not have any difficulties to breathe. We had one aortic bronchial fistula directly after the operation. Of course, this was an urgent operation, reoperation, but the patient is still fine. We had one venous thrombosis, but this was 1 year after the treatment. So we do not really see a relationship between this thrombosis and the APO treatment, which increased, of course, the hematocrit. And then we have one, I did not show here, I'm sorry that I'm not showing here. We had one patient who died on the second day post-operatively, a 73-year-old guy. He died from severe and fulminant pulmonary embolism. I mean, we do not actually see the relationship between the graft and the death of this person. Of course, it's related to the operation itself because he is not mobilized in the first 2 days. But the hematocrit was completely normal. There was no sign of side effects of the erythropoietin. So we believe, and the pathologist as well, that this was just due to the operation itself, but not specifically to the trachea transplant. In total, we have a median of 60-month follow-up in all patients together, and the patency of 100%, except the 3 that get a biodegradable stent, but it's also patency, even if there's a malacia in the distal anastomosis. This CT I want to show nearly at the final of this talk is the follow-up of the first patient that we transplanted in 2008. The young lady, she received a left bronchial replacement. This was post-operatively 1 month after the transplant, and now she came in April for the CT, post-op CT, and it looks completely open. It looks very good. The 3D animation looks very good, and also here you can see it looks very like a native. So this is a native one, this is an engineered bronchi, so it looks very good, and she's doing very good. In conclusion, the transplant of the airway is feasible with and without immunosuppression, in our case without the immunosuppression, both in adults and children with benign and malignant diseases. The most important is this to avoid and inhibit apoptosis via this boosting therapy by high EPO level and by recruiting progenitor cells from the bone marrow by a GCSF. We are able to engineer the graft in vivo without any in vitro cell culture or in vitro cell seeding. So we skip the whole in vitro part. And this is, I guess, the most advantage comparing to the old method that we use. You can use it actually everywhere, and everybody can use this technique because it's so simple. As long as you have the graft and as long as you have a hematology department which can isolate the mononuclear cells. At the end of this talk, I just want to give a short summary of this first transplantation of using an artificial scaffold that we recently done in Stockholm in June this year. We transplanted the distal trachea with a carina in both left and main and right bronchi, and in a patient suffering from a primary trachea tumor. And we used a CT scan, and based on the CT, a polymer matrix was used to engineer the scaffold. That was done by a group from London. They are working with these polymers since several years, so they're very experienced in this field. And then we used actually the same method as I explained before for the native graft. And so this is an unseeded scaffold. It looks, it looks like a trachea in a white color. This one is engineered after seeding in a bioreactor for 2 days. We injected all these boosting factors in the same method as I explained before, and transplanted then in the patient, and also continued this boosting therapy for 2 weeks post-operatively in a systemic fashion. The findings coming very soon. I'm sorry that I could not show it here. Our current and future research projects are standardizing the trachea engineering, improves the concept, and especially the in vivo concept, and comparing to the synthetic graft. Then we are we're looking to heart, lung, esophagus, pancreas, and liver. This is all done by an international cooperation with United States, Germany. This one is a group from Japan. And then we are focusing on stem cell therapy, but mainly on mobilizing the autologous stem cells without any in vitro culture, to skip the whole problems of in vitro culture and re-infusion into the patient, which is a very, very big concern issue in this field. This is our group. Of course, the chief is Paolo Macchiarini, and this is all our collaborators. Thank you very much for your interest.
Thank you for that really fine, groundbreaking presentation. Are there questions?
Yeah, I have a question. So those new tissues formed, which is from a host or implant?
Sorry, the last part I didn't.
So those new tissues formed.
Yeah, the synthetic?
The stem cells is from host or endogenous stem cells or you implant cells?
No, it's autologous. It's autologous from the recipient.
Oh.
We took the stem cells from the recipient, who get later the whole organ.
Yes, you implant
Yes.
to the scaffold.
Yes.
Then you graft to the patients.
Yes.
Then they form the new tissues.
Yes.
These new tissues is generated by host, because you do the boost.
Yes.
Boosts are stem cells. Those stem cells also participate from the new tissues,
Exactly.
or just you implant cells.
No, we actually it's a combination. We seeding them. Sometimes we're seeding them before, but we also use the mobilization of autologous cells from the bone marrow, and we hope that these mobilized bone marrow stem cells or these progenitor cells, they're participating in the whole remodeling. So we expect that both cells are involved in the whole process.
What do you expect the difference to be between the decellularized trachea and the artificial polymerous trachea?
Yeah, very, very interesting point and very, very interesting question. For this case that we operated in June, it was the case that we really need the trachea transplantation. This patient, he had a progress of the tumor growth, even under radiotherapy and chemotherapy. And he's a young man, he was 36, so there was no time to wait for donor, and there was no time to decellularize the trachea. So this was like the only possibility for this young man. And of course, right now I would say, I would prefer right now the native graft, if there's time. Because the native graft currently has this pro-angiogenic factors, which of course is very helpful. Of course, the matrix, the extracellular matrix, still contains several proteins which are very important for the engraftment. However, we saw that if you use a boosting therapy, you can nearly make the synthetic graft became the same results. I mean, we had the same results after 1 week taking biopsies, for instance, from the graft. It starts to bleed, so it was impressive. It was really amazing, and we could see after 4 days the mucosa on the synthetic graft, which looks, of course, there was an inflammation, let's say, a slight inflammation, but there was mucosa. So even the synthetic graft is very, very promising, and I guess if you can add some boosting factors or some growth factors, for instance, with nanotechnology into the graft, that would be fantastic.
Who was the group you worked with in London?
With Professor Spelling.
Short question and short answer, if possible.
Short question. Great work. Now, were you able to trace the cells that you've seeded? You would think that the cells, when you attach it intraoperatively, the cells are not adhered to the scaffold, and they would not be vascularized. And considering the environment where it is very dry, and it would be contaminated, through the air. So were you able to trace
So-
these cells?
I mean, we didn't trace them. We didn't trace them in a patient, of course. We didn't trace them ever. And in animals, we just can say that the mononuclear cells were able to differentiate into chronocytes. So we could see the differentiation. We do not know if these cells that we actually seeded were differentiated or they just promote the engraftment, the homing of progenitor cells that differentiated. So this is also a concern for the next project for the next future.
Okay, final question.
Maybe it's just a bit naive, but I was curious. Have you ever tried in an animal model to put inside the trachea, the decellularized trachea, without the preceding?
Yes.
And does it work? It doesn't work. So there is the need.
Yeah, the need is not to put directly epithelia or directly chondrocyte. There is no need for that. But the need to put some cells. Because if you have no cells in the internal layer, then you have contaminations within 1 week or within 2 weeks. If you not put any cells on the surface, on the external surface, then you will get within 2 weeks fibrosis and the construction and that's the collapsing of the graft.
Thank you.
Okay, thank you very much.